Oggi è una puntata nuova e diversa rispetto a quello che sono i nostri standard. Avremo un ospite internazionale per discutere di una molecola importantissima, di un pathway importantissimo, quindi sarà una puntata dedicata più ai nerd della scienza, al nostro pubblico che è appassionato di scoperta, ricerca e che può avere anche un interesse sull'anabolismo, ma vedremo perché l'anabolismo, non inteso semplicemente in ottica fitness. Hi everyone and welcome back to this new episode of the LifeX podcast. I'm Manuel Salvadori and today I'm going to have a different setup for my episode. I will have a different co-host, which is going to be Dr. Nicolas Di Leo. Hi Nicolas.
Hello everyone.
And we will have an host that will be introduced by Nicolas. Nicolas is a researcher, a student, and also, we can say, a former member of the performance team as a nutritionist, a future nutritionist. So, Nicolas, let's get it to you.
Okay. Hello everyone. Today we will be joined by my current supervisor, Professor and Dr. David Sabatini. David got his MD PhD in 1997 at the Johns Hopkins University School of Medicine and is currently a senior group leader at the Institute of Organic Chemistry and Biochemistry in Prague. But he also leads a lab that is about to open in Boston, in the US. David, thank you for being here today.
Thank you, Nicholas. Thank you, Manuel, for having me.
So I think we should start. We should start by basically mentioning that you are one of the leaders when it comes to mTOR research in the world. You're one of the persons that contributed the most in this area of research. And mTOR is an acronym that stands for mammalian or mechanistic target of rapamicin. So we cannot talk about the mammalian target of rapamicin without talking about the molecule that gave it its name. So can you briefly share with our audience what's the peculiar story behind rapamicin?
Sure, Nicholas, happy to. So I don't know if I'm a leader or not, but I've certainly worked on it for a long period of time, and it's been my passion for a long period of time. So rapamicin is this very interesting molecule that has sort of an interest from a cultural point of view, particularly now because of these pro-lifespan effects, but also historically because it comes from Easter Island, which is the indigenous name is Rapa Nui, so it has a sort of interesting history to it. And then because it led to the discovery of mTOR and particularly of TOR, which is the yeast gene that preceded mammalian TOR, mTOR, or actually it's officially now called mechanistic TOR. It turns out you can't officially name proteins with mammalian. And so there's those two stories of mTOR and rapamicin are intimately tied. And without rapamicin, there would be no TOR or mTOR. So the first attempts to find out how rapamicin worked. So people had recognized that rapamicin had interesting effects. It inhibited immune cells. It inhibited yeast. So there was a lot of interest in figuring out how it did. And people like Michael Hall really pioneered using rapamicin to study yeast, and he eventually found mutations in genes that he called the TOR genes in yeast. People like me, Stuart Schreiber, Bob Abraham were on the mammalian side, and instead of using genetics, which is what people in yeast do, we actually use biochemistry to purify mTOR bound to rapamicin. There are some subtleties in that story, but it's not really worth describing it. So mTOR was discovered because it interacts with rapamicin. That's the bottom line.
Okay. So you're one of the people that basically contributed the most in the discovery of mTOR. But what has been the role of rapamicin? Like, how did you think about using it in your favor? And what has been the biggest challenges and aha moments during those days? If there is something fun that you want to mention.
Sure. So you're certainly going far afield. So we probably should take a step back and describe what rapamicin does a little bit.
Yes,
right?
Go ahead.
So rapamicin is approved as a drug, right? So it's used clinically in a number of situations. For example, in organ transplantations of kidneys. It's used in what's called preventing restenosis in the heart of blood vessels. it's used in certain rare diseases. Sorry, I'm getting now some feedback where I'm hearing myself. Before I wasn't. So that, it's a clinically useful drug, and like any drugs, has to be used carefully. A number of years ago, people started to recognize that rapamycin, again starting in yeast, but then in worms and flies, and then really what caused, I would say, the rapamycin mania that there is now came from work in mice, where it was shown that if you treated mice with rapamycin, they live longer. And many studies on lifespan and drugs or interventions, they can sometimes be hard to reproduce. The effects can change with different groups. These experiments were done very well in multiple sites and have now been replicated by many labs, including my own. And it's clear that rapamycin causes anywhere, depending on the gender, the strain, the age, somewhere between a 10 to maybe even 25% increase in lifespan of mice. Okay. So this was obviously very captivating to people. And, the people that were driving this, of course, recognize rapamycin is also a drug and it also has some side effects. But what was interesting is that they seemed to be able to use it at low enough doses that this was not so much of a challenge, right? And so this has led to quite a bit of interest in people using rapamycin, and there are many people that use rapamycin. In fact, the most common question I get is, do I use rapamycin? Which I don't. And should others be using rapamycin? Maybe even Nicholas asked me this question.
Yeah, actually, sorry, Nicholas, but in many forums or Reddits or subreddits, there have been a lot of people talking about rapamycin as an anti-aging agent. And actually, as many underground scenes that usually go, it becomes a little bit further from the area of study. So people start using that in a more free way or without having the background to understand what they really are doing. So maybe I just read on a forum or a subreddit that rapamycin, maybe I'm going to read 2 or 3 lines about how rapamycin is better for my aging, and without even knowing what I'm going to do or what I'm going to use, I start taking that. So it's a very dangerous way to share informations, in my opinion.
Yeah. And, and I think, Manuel, the question of, what you haven't asked me yet is, well, why does rapamycin have this effect, right? And I think this is intimately tied to information that as a community we've had for much, much longer, for many decades. And that is that if you reduce the amounts of nutrient that an animal takes, what can be called dietary restriction, caloric restriction, intermittent fasting, skipping breakfast, whatever. Anything that reduces basically an amount of calories that you're getting without causing malnutrition, right? Keeping vitamins and everything up. This has been known for literally decades that it prolongs the lifespan of many kinds of organisms, many kinds, much more than has been tested rapamycin. And there's probably an intimate connection between mTOR and rapamycin and caloric restriction in that mTOR senses nutrients, right? And so probably what rapamycin is doing is mimicking this normal response to caloric restriction. And so the real question that I think a lot of people who take rapamycin need to be asking themselves, A, are they taking enough? Because, low dose may not profoundly impact the pathway. But even more important, they already may be living a lifestyle that's inhibiting their mTOR, right? A lot of people who care about health, right, are already reducing the amount of food they take in. They're already exercising. And so it could be that they're already in a state of relatively low mTOR activity, which is what rapamycin causes. And so this is an unknown for us at this point. So.
Talking about mTOR, using a different point of view. So the person that normally is passionate about fitness or fitness enthusiast, a lot of our listeners are passionate about fitness, and usually it's interconnected in a way. So when people are passionate about nutrition or anti-aging, they're usually passionate about exercise or physical fitness. And we are kind of used to link mTOR to anabolic states. In the introduction that I did in Italian, I said that we are going to be talking about anabolic, but on a different levels. So we're always used to understand, or to when you get a supplement, a normal supplement for the gym that is advertised, usually the name mTOR comes up, stimulating mTOR pathways, the greater stimulator of mTOR in order to grow your muscles. So listening to you, actually, we can understand that mTOR is linked to a whole different level of understanding of the physiology, of the human physiology, sorry. So can you explain a little bit further how much mTOR is not only linked to looking jacked or creating muscles or getting bigger?
Sure. So look, if you think about some of the most basic things all organisms have to do, is they have to identify food in their environment. They have to understand whether this food is of good quality and is appropriately balanced, and they need to consume it, and then they need to know that they consumed it, right? This is like as basic as it gets, right? Now we kind of forget that because we live in an environment where there's a lot of food, right? For most humans, unfortunately not all humans. And so food abundance is our life. Food scarcity is the life of most organisms that live out there.
Mm-hmm.
Right? Mm-hmm. And so it's not surprising then that we have a system that can detect food abundance and therefore change all our physiology. And I think you already appreciate that if you're in a lower food or a prolonged fasting state, everything changes, right? There's no aspect of your physiology which is not impacted by that state. So that is why when you inhibit mTOR, which mediates a lot of that response to low calorie states, you have many impacts on physiology, right? It's not simply the muscle. And so you end up in this sort of conundrum where mTOR is good in some situations like the muscle and is probably less good in other situations, for example, like the liver. And this brings up one of the major challenges of how to use either food, like you alluded to these supplements, a lot of them contain leucine or arginine.
Yeah, totally.
Like these are amino acids that activate mTOR that we figured out sort of the mechanism by how they do this. Those are natural ways of activating. Rapamicin is an unnatural way of inhibiting. And so the problem with all of those tools is they're affecting mTOR in all your tissues, right? In some ways. And so there's not an easy answer to how you exploit mTOR for your benefit in some tissues and not others. And as Nicholas and I talked about, right, you want to take supplements to build up muscle. And I think it's been quite well shown, and you guys probably know better than I do, that does work, right? Protein supplements do seem to drive muscle mass. On the other hand, you want to inhibit mTOR for a longer lifespan. So, where do you land between those two things, where you're promoting mTOR for, as you called it, getting jacked, but you want to inhibit mTOR for prolonging lifespan. So, okay. I have ideas about that, but those are very unsolved problems now.
Okay,
and
perfect.
We said that like Manuel is a nutritionist, and I sort of am as well. And most of our listeners are, again, passionate about nutrition research. So you sort of hinted about this question that I'm about to ask in the previous point. But since you're also interested in the nutrient sensing capabilities of mTOR, can you give us an overview mechanistically on what mTOR is sensible to? Because most people know leucine, but there are many other things that mTOR is sensible to. And how does mTOR detect these other compounds? What molecular complexes are involved? Because mTOR is not alone, right?
Sure. So I've been most interested in this question, Nicholas. What does mTOR sense? What does it detect? And how does it do this? And the answer to that, and I think this is the reason that mTOR research has been so interesting, is that mTOR detects almost everything. And of course, this makes sense too if you think about the first statement I made. Organisms need to detect lots of things about their nutritional state to make decisions, right? Think of the different kinds of nutrients. You have very simple ones like oxygen, right? You have carbons like glucose. You need nitrogens like amino acids. You need some lipids, right? You of course need micronutrients and vitamins, and you need certain minerals. And mTOR has ways of detecting almost all of these. You need to know your overall energy levels, right? So ATP, surrogates of ATP. And so what does mTOR detect? Well, most of these things. Now, I do think there are tissue-specific mechanisms that make some more important than others. So, for example, in the muscle, I think the amino acids are quite important. And there we have discovered these proteins. One is called cestrin, for example, that directly bind the amino acids and transmit the signal to mTOR. But in many tissues, the way mTOR is regulated is in a coordinated fashion by insulin, right? So when you eat, particularly carbohydrates like glucose, you secrete insulin. Insulin goes to many of your tissues, including the muscle, including the fat, including the liver, and says, look, we have nutrients. Take them up. Do things with them. So mTOR has really two masters in a big picture way. It has the hormones that are secreted, like insulin, by the pancreas. And then it has the individual nutrients, like amino acids and glucose and lipids and oxygen, that at each cell can be detected. Because if you're a cell in a multicellular organism, you have to be in tune to what the rest of the body is doing. But you also have to think about yourself. Like, do I have the things I need? And is my body, where I live, in the right environment for me to grow and to do anabolism? So there's lots of different inputs. They vary in different tissues, and some of that, frankly, we just don't have the answers to. We have the molecular mechanisms at the level of individual cells, but how those work in different tissues and different situations, we don't know.
Okay, and do we have evidence regarding possible dysregulations that can occur in disease states in people that have diseases like cancer or metabolic syndrome? Do these amino acids or energy or all these inputs signal differentially, in healthy versus unhealthy patients? Yeah,
so there are many connections of mTOR to disease. You mentioned some. I mean, cancer is one of them. Certainly metabolic syndrome, there's been an argument that sort of this hyper mTOR activity that you're getting in many tissues is detrimental. A very interesting one is neurodegenerative diseases. Many neurodegenerative diseases are associated with dysfunctions of lysosomes and autophagy. I'm sure your listeners know autophagy, right, is where you break down your tissues in lysosomes. So there are many connections. That being said, mTOR inhibitors in some specific cases have had profound impacts, but are they, for example, a very general anti-cancer drug? Probably not. And the reason is simple, is that our normal cells also need mTOR, right? And so the therapeutic window between hurting a cancer cell specific and hurting the rest of the proliferating cells in our body is relatively small, unless the cancer cell has a very specific mutation that is very specifically turning on mTOR. And there are some of those. There's genes called the tuberous sclerosis complex genes that are involved in cancer and rare disease. And there, the effects, there's better therapeutic index there. So it's a, what's the expression, right? It's like, you can't have too much mTOR activity, but you also can't have too little, right? There's sort of this perfect in between that, and it's hard to thread that needle. I'm not sure what the Italian expression is for that. But.
Mm-hmm.
Okay. And I'd like to go, before going on the clinical side, I'd like to get a step back because when trying to we always try in our podcast, in our episodes, we always try to get practical insight from the research. So there is one question that, putting myself into the side of a fitness passionate or even a nutritionist, a sports nutritionist, as I told you, we always get the question or get the advertisement of how stimulating mTOR is good for you. And you just explained how the picture is more complex, a lot more complex than how it seems. But I'd like to point out one thing, one point of view. Is it possible, is it correct to say that it's possible to stimulate mTOR just by a single element. Let's say I take leucine and I get more mTOR stimulation. So looking from my point of view, it's a bit of a reductionist point of view. Like I do this and I get this response from my body. So I take BCAAs or branched chain amino acids and I get more mTOR stimulation, or I get protein, I get more mTOR stimulation. Does it work like this, or again, we can say that it's more complex than this?
Well, so Manuel, of course, as you realize, it depends when you're taking these amino acids, right? Are you taking them on a fasted state where you haven't eaten for some time? And, to really fast a human, you need, it's not just an overnight fast, right? You can take a mouse and in 12 hours, it's in a very fasted state. It starts to lose weight. In 24 hours, it's extreme. 48 hours, it's like not happy. And at 72 hours, it can be dead, right? A human, it takes some time to really go into a profound fasted state. So I would say it depends on that, right? So typically, if you give a mouse that's fasted just leucine, you will get a little bit of insulin secretion, right? Leucine can cause some insulin secretion. And that can be enough where if you think about what I told you, mTOR has two masters, the hormones and the nutrients. And it's think of it as in computer terminology, an AND gate, right? You need two inputs to go down, right? You need two pluses to send a plus down. In that state, you can get enough insulin that you can then activate mTOR in tissues, for example, the liver and in the muscle. You can certainly get that. I think if you're in a very, very low insulin state and you take an amount of leucine that causes no insulin secretion, it's going to be harder to do that. So I think the simple answer is that in your physiology, unlike what I can do in a cell, right? I can put a cell in a dish and I can remove all the glucose. I can remove all the insulin. I can't do that to you, right? Even when you're fasted, your glucose is at a pretty reasonable level. And so I think in general, yes, individual nutrients can activate mTOR. Depends a little bit what state you are in, but you can't think of them as individual nutrients because your body has nutrients, right? So things like glucose you're making, even amino acids, right? You can't lower your amino acid levels that much because as soon as you do, you break down your muscle to release amino acids, right?
Yeah, for the pool.
So it's a little bit of a different situation than what we can do in a tissue culture dish.
Okay, thank you.
And you mentioned that something that I always found interesting, and I want to hear your thoughts about is the amino acids and proteins can also spike insulin. And I always wondered why that is, like why evolutionarily not having glucose. For instance, if you take whey protein, okay, which are like 80%, 90% protein and basically zero carbs, you spike your insulin massively, right? And so I was like, why would, how does that work? Why would we need, insulin to be spiked that much if there are no carbs around? You know what I mean?
Well, I mean, look, I think, Nicholas, I'm not an expert in beta cells or insulin signaling, but I think if you think about insulin being a driver of anabolism, obviously, leucine and other amino acids can drive anabolism. Of course, you can also do gluconeogenesis, right? And the backbones of some amino acids can be involved in making glucose. So if you think of it from like the big picture point of view, are you providing nutrients that can give a lot of the functions that the cell needs? It kind of makes some sense. Now, the mechanisms by which this happens, it turns out the sensors that we've identified are in beta cells. And there clearly is crosstalk between the glucose sensing and the amino acid sensing. So mechanistically, we can understand. On the other hand, leucine by itself is a very poor insulin secretagogue compared to glucose. And it really, and from what I remember, it really doesn't work unless there's a little bit of glucose around. It can potentiate, but it can't substitute. And so even the whey protein that you're saying, it has some carbohydrates in it, right?
Yeah, true. There are like very few, like 1 or 2 grams of carbohydrates per 100 usually. But
sure. That's not nothing, right? It's
okay.
Yeah, sure. And we mentioned about diseases earlier, and I was wondering, like a very broad and just food for thoughts question: how do you see the future of mTOR targeted therapies evolving? What are the most promising areas of mTOR research now and in the future, because what we are doing even now in our lab is trying to find mainly small molecules to inhibit mTOR. But there are some other strategies, especially in the longevity field, like gene and cell therapy, epigenetic reprogramming. And how do you see like all these fields, competing with each other in 50 years' time? Or like, okay, let's say 20, let's be more.
All right, well, let's take those a little bit. Let's break them down into manageable chunks, because obviously you asked lots and lots of things in that question. So if you just look at mTOR itself, right? So one very profound question is, are we getting the optimal mTOR modulation for health and lifespan, right? Is, for example, rapamycin the best we can do? I don't think we have the answer to that. And my feeling is that if our target is mTOR itself and rapamycin is directly inhibiting mTOR, rapamycin might be the best we can do. It might be, because if we make inhibitors, and rapamycin, just so everyone knows, is a partial inhibitor of mTOR. We've shown that a number of years ago. It's a partial inhibitor. If you knock out genetically mTOR, your cell is dead. Rapamycin does not kill cells. So that already told us that, mTOR has rapamycin resistant functions. But you can make better inhibitors that inhibit all mTOR functions, and now cells are extremely unhappy. Animals are extremely unhappy. So if mTOR is your target, rapamycin might be as good as it gets. It might give you enough inhibition to have an impact, but not too much to cause a lot of toxicity. But as you know, Nicholas, because you've been working with us now, I do think there are interesting targets upstream of mTOR. So the nutrient sensing pathway, we originally thought was dedicated to mTOR, that it was a very linear pathway. Nutrients, sensors, mTOR. What we now realize is that nutrient sensing pathway actually talks to many other proteins besides mTOR. mTOR is probably the most important, to be fair. But so if you really want to get closer to mimicking the impacts of lower nutrients, you really can't go after mTOR itself. You have to go things upstream. And so I think that's one very promising opportunity. And that's something we're interested in looking at. Now, it's hard because the targets in that pathway are not like mTOR, which is a kinase, which we know how to inhibit kinases. Some of these other proteins we don't know how to inhibit. So that's the mTOR space, and I'm excited to try to go after that. I think the other area in mTOR that is interesting is thinking about tissue-specific impacts, right? What if we could just activate in the muscle? What if we could just inhibit in the liver? What if we could just actually impact the brain at certain times, right? Not cause depression, but maybe protect against Alzheimer's, which there's some evidence that might happen. So tissue specificity is the other one. Now, rapamycin is a very, as we say, greasy hydrophobic molecule. It crosses all membranes.
Okay.
So there's no real specificity of rapamycin. If you look, rapamycin goes everywhere. So can we start to think about modifying it so that's not the case? I think so. I think there are things, using antibodies, for example, or modifications that we could go down that road. And so if I was sort of a dedicated drug developer, those are the two avenues I would go to. Upstream of mTOR and then, tissue specificity, and which could have implications for disease as well. Now, you brought up all these other things like reprogramming with Yamanaka factors, right? And so how does that compare to mTOR? And to be honest with you, I don't know. This is something I've always been interested in, is what is that connection? And it really goes down to a question, Nicholas, that I've brought up to you, which you don't seem very interested in, is fundamentally, what does it mean for a cell to age, right? Like this whole aging problem, which we all agree is a problem, I don't think is solved or we make any significant contribution until we understand what exactly is going on. And there are very few people trying to figure out exactly what's going on. So what is it about reprogramming that's helpful? People, are, trying to reprogram this way, small molecules, viruses carrying the Yamanaka factors, alternative factors, at young age, people are doing all of that. But there are very few people asking, what is actually happening at a deep molecular and biochemical level and ultimately cellular level? To me, that's the central question. And then if we have insight there, you have to ask the same question about mTOR and rapamycin, which you guys didn't ask me. A very common question is, well, why does rapamycin work? Right? And I have some ideas and there's some evidence, but how are those tied? Right? Will there be synergy, for example, by doing reprogramming plus rapamycin? I don't know. Maybe. I think probably there will be, because I don't think they're fully overlapping. But we don't have answers to these types of questions because we fundamentally do not know what happens. Right now, the literature suggests everything breaks. That's a very unsatisfying answer for people who want to fix something, right? Because when everything breaks, you typically throw away that thing and make a new one, right? Like everything broken in a building, very few people take the effort to fix everything in that building. And so I think we have to do a much, much more work at the fundamental cause of aging. That is, to me, the big question. Is there a fundamental cause which then leads to everything? Or is it just entropy? Right? Okay. And everything is falling apart at some rate.
And what do you think should be the research that needs to be done in order to answer this question? You said that very few people are working on this. Maybe some of our listeners would be like, okay, I want to start working on this, but how? Where should I start? What should I do? You know what I mean?
Yeah. So I don't fully have answers to that, Nicholas. And it's not that other smart people besides us have thought about these kinds of questions. And so I think at the most basic level, we need to be able to. So if I had a dream experiment, I would want an old cell and a young cell. And I want to know every parts list down to individual atoms. This is what I want to know. Every atom that's in there, in every polymer, with every configuration. I want that very, very deep part list to be able, because the first thing you have to do to understand something is you have to describe it. Right. And so we first have to be able to say what's really fundamentally different between an old cell and a young cell, and then obviously building up the complex. So that's where I would start with. As you know, Nicholas, I'm also interested in, why rapamycin works better if you give it to younger organisms than older ones. Even if you give it intermittently, it's clear that rapamycin given to a young mouse for a period of time has a bigger impact than given to an older mouse for the same period of time. Why is that? That's a pretty fundamental question too. But I admit that these are not easy questions to solve. And moreover, where lifespan is your output, they're also long experiments to solve because you have to let mice age, right?
Yeah.
Looking at it from another perspective, you say that correctly because you're a scientist and you know your boundaries and you know what your very area of expertise is, but you have a lot of culture about different aspects of even outside of your specific area of interest. And you said you have no answers to that. But if I ask you on another point of view, your questions. So which are the questions that you would ask about longevity research? So you say no answers, but I'd like to ask you if you have one or two or three specific questions that you would like to be answered. Because if I read a book recently on anti-aging, and of course rapamycin has been explored, has been named, but talking about how cells get senescence or how to get older, from my understanding, it was pretty clear the 8 different theories of why cells get older and why we get older and how we get older. It looked like it was a very certain subject. It was full of conclusions, not full of questions. So just by that book. So it's a research book, but just one book.
Which book is this, Manuel?
I will tell you later because I have to translate that in English. So I'm not familiar to doing that instantly, but I will tell you. So what are your questions about this subject?
So, maybe it's possible that I've missed some huge insights that have happened in the last few years, but I don't think so. Many of my good friends are in the aging space. Mm-hmm. So again, I hinted at this with the response to Nicholas is I would like to have this sort of very, very deep parts list of an old versus a young cell, and really understand exactly what is different between those two. And I'd like it, of course, over time, right? Not just a very old, decrepit cell. And I have to look at that, right? And understand, is everything are all the proteins slightly misfolded and oxidized? Is the DNA have many different mutations? Or is it, something that really starts the process? I think a very, important point is there something that comes first? Right? For example, if I had to pick something, I would say it's DNA damage. That's the only thing that I see as a sort of universal connector between different kinds of organisms and cells. And I mean from very simple to animals, not between us and mice. Obviously, the potential DNA damage is similar. So that is, is there an instigating event, or are we talking about many parallel processes at once? If it's an instigating event, then, okay, we can intervene. If it's many things, it's harder. So that's one question. A second one, and I think I've mentioned this to Nicholas too, if you had to pick to me the most maybe feasible and interesting question, it would be, why does a human being live so much longer than a dog? Okay. They're not that different in size, particularly big dogs, similar environments, similar foods, similar genomes. Why is that? To me, that's a very interesting question. And I would say that the answer is encoded in the genome. Okay. So the genome of the dog versus the genome of the human must provide that answer. I think that's a pretty reasonable thing. I don't think there's some unknown virus, for example, that dogs have that are, making them live longer than we. There could be, of course, but given our similar environments, I would say no. So that is the question that I would want to number one. Pick. So those two in the aging space is what I think you asked me, right?
Okay. The book was Epigenetic and Endocrino-Immunology, Two Phases of a Revolution During the Science of Life, something like that you can translate.
Mm-hmm. Who's the author?
Bottaccioli Francesco.
Okay. I haven't read it. So the epigenetics, which of course is the modification of sort of the genome itself and genome activity by non-sequence changes, right? So features that are either proteins or modifying the DNA. That is an interesting idea that is what goes wrong over time. And basically it's like the software, the code is still there, but the reading of it goes wrong, right? Of the DNA code. And so that's a potentially interesting idea. I don't, it's not as universal as I would like. Some organisms really don't do sort of epigenetic control nearly as much as ours do. But it might account for why reprogramming works.
Okay.
Or at least works to some extent.
Going on let's go on an end note and on a more practical side of the research. Again, on longevity, you mentioned fasting, and fasting is a giant trend in the fitness industry. Also now in the nutrition field, people talking about different kind of fasting, 16 hours, 18 hours, 1 day per week, 5 days per week. It's a whole lot of difference, but is there a way we can explore, or we can identify the diet patterns that are most that can affect mTOR more substantially, so more on a more direct way? Is there a way that we can? You said, for example, something that to you it might be obvious, but not for our listeners. For example, when talking about an overnight fast, we're not starving the body. This is not obvious, and it's very important for our listeners. Is there some kind of insight on that field?
Yeah, so Manuel, this is a very interesting question, and this is a question that my friend Peter Attia and I have talked about a lot. Okay, what could we measure relatively non-invasively, of course, in a human being or in an animal that tells us that we've modulated mTOR, and ideally in tissue-specific ways for the reasons we talked about before? So the simple answer to that is no, we don't have this. And I think this is a major hindrance in the field. And so you asked me interesting questions, and I told you very basic questions because those are the kinds of questions I like to think about. But if we want to think about practical questions that I think we can think of solutions to, this is one of them. What are the things that we should be measuring in an intact human that says this is the activity of these pathways? Now, I have some ideas around that as well that are more technical, and I know I'm happy to go into them at some point. But we need to solve that problem if we are going to then apply scientific rigor to different diets, right? Because as you said, now people say, okay, well, you need to fast at least 18 hours to induce autophagy. And we know autophagy matters for lifespan because someone did an experiment in a worm. Okay. And I agree. Let's try to get autophagy activated. But do we really know we're activating autophagy without taking a muscle biopsy? No, we don't. Okay. So we need markers of these different states in humans to be able to make rational decisions. And I think there's a very interesting, even commercial opportunity to do that. You're probably still going to need a blood test. You know, ideally it'd be sort of like a breathalyzer type of situation, but we're probably going to need a blood test to do that. But I think that's feasible. I'd like to contribute to that, and I have ideas about how to go about that. But we need that. So the answer is, I don't know. People do all these things, and some work for others, and some, you know, and obviously people have different physiologies and different bodies and different food preferences. So it might be that many of these things work, but we don't have a scientific basis to evaluate them because we're not measuring anything to evaluate other than some long-term health impact, which you can't do experiments like that, right? So.
No, of course. So thank you for pointing that out. I think that's very important. So people, we can just, we need to accept just the fact that people are going to experiment with that. Everyone will go, apart from experimenting on a scientific field, but people will experiment on themselves. Someone might feel better with a kind of approach. The point is maintaining some key points, some key factors in their practice to make it logical at least.
Well, and having some metrics. So look, I know some people in the fitness field more body weight, you know, weightlifting than necessarily fitness. Although, as you said, these are very overlapping people, right? I'm always very impressed with how rigorous they are in what they apply and how they can understand what's going on in their body, sometimes even without measuring them, right? People can look in the mirror and know their body fat percentage, right? Based on how their muscles look. I've always thought that there's an opportunity to try to take that community and really try to integrate all that data for insights. So you can imagine there's a lot of data out there, right? Totally. That even you could apply in sort of machine learning type approaches to get at some of these answers. I think there's also an opportunity to do that because you're talking about a community that's rigorous in the way it does things and the way it measures the things it can measure, right? Yeah, totally. Obviously not measuring mTOR activity in the muscle, but it's measuring other things. But it does it in a way that's quite rigorous. So, you know, those are things I'd like to get involved in, how you actually organize. You need large numbers of people, right, to be reporting. But as far as I know, no one's systematically trying to collect that data. I imagine it exists on forums and you guys talk, but to really look across all these experiments that people are doing on themselves and collect that data, I think is an interesting thing to try to, how you do that.
I think that, sorry, Nicholas, if I go on further on that, but one of the greatest example is intermittent fasting, the 16-8, the approach, which was originally called lean gains. Nicholas, I'm sure you're familiar with that. And it's the example, a very good example of what you just said. So people starting first and foremost from the forums, from the studying on themselves, getting the point of 16 hours of fasting and 8 hours of refeeding. And then later, the scientific field came up going to study that because there were a lot of data of people collecting on themselves. So, for example, 2,400 calories in 8 hours, respect to consuming that in 12 hours. And I looked more jacked. So a lot of people were experimenting on themselves. A lot of data were present on the forums, on the Reddits, and some scientists, for example, Antonio Paoli from Italy, said, okay, let's test it. Let's try and analyze the science behind that. So starting from experience, they got the theory behind that.
Right, but I wonder, Manuel, if we could scrape that data automatically, right? The machine learning people, they go and scrape data off the web, off of databases, and they integrate and they organize it. And so I've always thought it'd be really interesting to, go into what people are reporting and accumulate all that data as much as possible and try to analyze it. And then, of course, it can lead to experiments, but sometimes it can be so convincing that maybe it's not even necessary. So.
Okay.
Like I always found extremely interesting when I first joined, I first started doing academic research by working on muscle mass, okay, muscle metabolism, muscle physiology, okay, and I noticed that people doing this kind of research were not actually interested in muscle mass. Like they were not going to the gym, they were not training. In my first internship at the University of Copenhagen, we were doing research on protein intake. And these researchers didn't know how much protein they were eating, or I was doing, research on diabetes, in Stockholm, and the researchers didn't know their fasting blood glucose. And I was like, it would have been so much more, so much interesting if, these the people that are so thoughtful about their bodies and about their health, or at least about the matter that they're researching, were at the same time the researchers. You know what I mean? It would be.
Yeah, I mean, I know some of those people that do both, but most people's working on building cars are not racing Ferraris, right? So it's I don't know. It's just not. It's people have jobs and they're and it's hard to, let's be honest, it's hard to keep up, right? That kind of intensity. And most people that I know that continuous glucose monitors, track everything they eat, blah blah blah. They don't do it they do it sometimes for years, but not for 5-10 years.
Yeah, totally.
Okay, I think on this note we can conclude. We touched on some history, some molecular mechanisms, some also clinical and physiological insights, and we gave a lot of food for thoughts to our listeners. I'm sure they will enjoy this kind of a different episode of the LifeX podcast. And I would like to thank you a lot, David, for coming here.
Thank you for inviting me.
Thank you a lot, even from me, for being here and for taking your time to answer all these questions. And again, some of the things that you said that were maybe obvious to you, I'm pretty sure that will be groundbreaking insight for people listening. So thank you.
Well, thank you both for inviting me. And I look forward to seeing it when it comes out in Italian, I guess.
Yes, it will be in Italian translated. So thank you to all our listeners. And again, if you are if you're happy with this new kind of format of LifeX podcast, just let us know, and we will provide more episodes like this. Thank you all for listening, and see you in the next episode.
Okay, bye bye.
Bye bye.